Continuing on in our study of genetics, we're going to go now to dihybrid crosses and exceptions to Mendel's laws. A dihybrid cross is when you're looking at two different traits at the same time. So let's give, for instance, of a tall red-flowered plant crossed with a short white-flowered plant. So the tall red-flowered plant, true breeding would be big T, big R, okay? And the true breeding short white plant would be little t, little r. Their offspring would all be heterozygous for both traits, big T, big R, little r. And I'm going to indicate the red here with the color red to show you that when that is present, it's going to be red. Okay, so if you cross two of those F1 offspring, then you would have what's called a dihybrid cross. We have two hybrid traits that we're looking at comparison of. So you make a Punnett square. This Punnett square is going to be bigger. It's going to be 4 squares across and 4 squares down, so a total of 16 squares. You will write in your parental gametes for each one. Each gamete is going to have one copy of each, one of the combinations, okay? And the way you do this is the same way you multiply binomials in math using the FOIL method. You get first letter of each pair, the outside letters, the inside letters, and the last letter of each pair. And so here we have the first, that's big T, big R, the outside, big T, little r, the inside, little t, big R, and the outside, little t, little r. I mean the last, little t, little r. So we have both of those the same way for the parents. And then when we do the Punnett square, what we're going to end up with is something like this, okay? So we have here, looking at the two traits that we're following, we have a total of 16 possibilities here. Now, you might notice that some of these are the same. For instance, each one of these right here has the same genotype. So you have to count up the genotypes and the phenotypes to figure out the ratio. So take a minute, pause the video, and count up how many of each genotype and each phenotype that you have. The phenotypes are going to be tall red, tall white, short red, and short white. And you can look at the different genotypes and figure out the different genotypes. So pause for a minute and write those down. All right, so you should have ended up with a phenotype ratio of 9 tall red, 3 tall white, 3 short red, and 1 short white, 9, 3, 3, 1. When you do a true dihybrid cross, like the one we just looked at, your phenotype ratio should be 9, 3, 3, 1. 9 with the showing both dominant traits, 3 the first dominant, the second recessive, 3 the first recessive, the second dominant, and 1 both recessive. The genotype ratio is a little bit different, okay? You have to write all these genotypes down, but you'll also notice that it forms a pattern as well. We have 1 to 2 to 1, where you have a homozygous dominant for the first trait, and then homozygous dominant, heterozygous, and homozygous recessive for the second. 2 to 4 to 2, heterozygous for the first trait, homozygous for the second, heterozygous for the heterozygous for both, and then homozygous recessive for the second. And then 1 to 2 to 1 again, with homozygous recessive for the first, and homozygous dominant for the second, heterozygous for the second, and homozygous recessive for the second. Now, here's a practice cross for you to do, okay? Talking about human traits. Attached earlobes are recessive to free earlobes. So free earlobes would be the capital E, and attached would be little e. And right-handed is dominant to left-handed. Right-handed, big R, is dominant to left-handed, little r. So show the parent genotypes that would be dominant and recessive on both of those traits. The genotype of the F1 would be big E, big R, and then show a cross between two individuals with the F1 genotype. You do that cross on your own, and you can check with us in class to see if you came up with the right thing. It should be similar to what we saw with the flowers on the flowers and the height on the previous slides. Now we're going to talk about some different kinds of inheritance that really don't go along with what Mendel studied. Mendel was very lucky in the traits that he chose to study because each of those traits was a true breeding trait, and each one was found on a different chromosome. And so he didn't really have any uh, differences. But we've since then, in years since then, other traits in other plants and other organisms have been studied. And so you have some variations or some exceptions to Mendel's laws. One type of exception is called incomplete dominance. This is when you have two different traits, but neither allele completely dominates the other. Normally, when we're writing letters, we'll show this with a, like for instance, the C here stands for color, and the R stands for red, and the W stands for white. And so when you have a true red plant and a true white plant crossed with each other, you'll end up with a heterozygous individual that has a red and a white, and those flowers are going to be pink. Now, this is not in pea plants. This is in some other kinds of plants. I believe this is the snapdragon, but you're going to notice it's slightly different thing here. So you will be able to see the 2 to 1 ratio that you normally see just in the genotypes would also show up in the phenotype because these neither allele completely dominates the other. What you have here is a blending or a mixing of the traits. Here's another example. In carnations, you've got red and white carnations producing pink. And here we have some kitty cats, a black cat and a white cat producing gray offspring. So in an incomplete dominant situation, you have a mixing of the traits or a blending of the traits together. Another example is called codominance. In codominance, both alleles are expressed. And here's another example with cats, okay? If you have a white cat and a black cat here that are mixed with this particular trait, you'll end up with a cat with spots of both different kinds. We'll talk about some cat inheritance later on, but that's when you have both alleles are expressed. Here's an example of a horse. This is a horse called a blue roan, and it's not really the gray color, but if you look at it closely, it'll have both white hairs and black hairs. This is a heterozygous codominant trait, okay? When you cross a black horse and a white horse of this particular strain, and you're going to get the mixing, the codominant characteristic of the blue roan. In order to find out what the genotype of an organism is, sometimes you have to do a test cross. This is when you're trying to figure out whether when it has the dominant phenotype, but you're not really sure what the genotype is. You can cross it with a recessive known genotype to find out what the genotype of the original parent was here. And so if you end up with all purple offspring here, then you would know that this was a homozygous dominant genotype. If you end up with half purple and half white, you'd know that this was a heterozygous genotype. Here are some practice problems for you, also involving cats, okay? We have long tails, short tails, and no tails. Okay, long tails is a dominant trait, homozygous dominant. The heterozygous gives you a short tail, and a homozygous recessive gives you no tail at all. So this would be an example of, which one do you think it is? This is incomplete dominance. Okay, so you need to do Punnett squares showing these traits, and you can check in class to see whether you came up with the right answers on these. The genotypes and the phenotypes and the ratios. All right, another exception to Mendel is when you have multiple alleles. This is when you have more than two alleles in a population that control a trait. Now, a lot of times in this case, you have only two alleles in each individual, but there are multiple alleles in the population. And an example of this is human blood types, okay, the ABO blood groups of humans, okay. If you have type A blood, that means you have the A allele. If you can be homozygous for the A allele, or you can be heterozygous. If you have type B, you have the type B allele. Again, you can be homozygous or heterozygous. If you have both the A and the B, they're both expressed. They're codominant, okay? And so they're both expressed, and they're both dominant to the recessive O allele. Now, most of the time, if you're doing real genetics problems, you'll use the letters with the superscripts here. Oftentimes in class, we'll just use the A, B, and O just because it's easier. But this is the way it's really expressed when you look at real genetics. And we'll talk some more about blood type in class. Another exception to Mendel is polygenic traits. Polygenic traits are traits that are controlled by two or more pairs of genes. An example of this is skin color. In humans, you can see, even just looking around your classroom, lots and lots of different colors of skin. That's because there are three pairs of genes that code for skin color, and they all combine together. If you have all dominant genes, then the skin color is very, very dark black. If you have all recessive genes, then it's very, very light white. Now, this is not the same thing as albino. That's a totally different thing. We'll talk more about the albino later on. But if you have three pairs of genes that code for it, then you can see that if you have the more dominant genes you have, the darker your skin color will be. The more recessive genes you have, the lighter your skin color will be. That's called a polygenic trait. And skin colors like that, hair color is somewhat like that, eye color is definitely like that. And we'll talk about some more of those in class. There are also, another thing that Mendel didn't have is something called linked genes. These are genes that are located on the same chromosome. Now, actually, in Mendel's case, there were two different traits that he studied that were linked on the same chromosome, but one was at one end of the chromosome and the other was at the other end of the chromosome. And so the crossing over that occurs during meiosis occurred often enough that it didn't really have an effect on the inheritance of those traits. But linked genes oftentimes are inherited together. Um, and we'll talk some more again about linked genes in class. The main example of linked genes that we talk about in our study of genetics is something called sex-linked genes. These are genes that are located on the X or Y chromosome. Now, the Y chromosome, remember, is shorter than the X chromosome. It has fewer genes, and none of the genes that are on there will match the genes on the X chromosome. A single recessive gene on an X chromosome will be expressed in a male because a male has only one X chromosome. Females, they're less likely to be expressed. They'd have to have two recessive genes, and so the recessive trait is not going to be expressed unless there are two of the alleles for that trait in the female. Examples of sex-linked genes are color blindness and hemophilia. Color blindness is the inability to distinguish certain colors from each other. The most common kind of color blindness is red-green color blindness. It's individuals with this kind of color blindness have difficulty telling the difference between red and green. They both look kind of brown to them. And hemophilia is a condition where your blood clotting factor is missing. And so these are called bleeders. If you get cut, you can bleed, sometimes to death, depending on what kind of hemophilia you have. And if you've already studied the Russian Revolution in world history, you may have learned about the Crown Prince Alexei, who was hemophiliac. And the royal family of Russia tried to hide that fact. This concludes the notes on the exceptions to Mendel, and we'll talk more about gene mapping and about gene expression in our next notes.